A very good afternoon to everyone. On behalf of DDH 2020 training team and Director CSAR NEAST, Dr. G.N. Sastry, once again we welcome you all for the Vertical 1 session. Today we have eminent speaker, Professor Ramarao, former Director, Ramarao Poduri, to deliver the lecture in Vertical 1 session. On behalf of Dr. G.N. Sastry and our training team, we record our sincere thanks to you, sir, for accepting our invitation. Professor Ramarao completed his PhD in 1986 from Banaras Hindu University. He has substantial experience in working both academics as well as industries. He has more than 40 years of research experience, research and teaching experience. His area of interest are understanding the receptor mechanisms in normal state as well as the disease state, and tolerance and dependence, regulatory toxicology. He is the chairman and committee member for various drug and pharmaceutical research program of our Indian government as well as various pharma industries. He has 9 patents to his credit and more than 130 publications with a touch index of 40. With a small introduction, I request Dr. Ramarao to deliver the lecture on drug discovery and development on COVID-19. Sir, over to you.
Okay, good afternoon. Thank you very much for the nice introduction. This is a drug discovery hackathon 2020 is a pioneer program initiated by CSIR and other actually organizations of government India. So, the drug discovery is one of the very important aspects, and my talk is in continuation with Dr. Sastry sir and Professor Bhartam on basics of drug action. I'll be talking mainly. May I have go ahead, next slide, please. Next slide. So the challenges of new drug treatment paradigm, whenever we talk about it, so this is actually the symptomatic treatment, stop disease, and the disease prevention as well as the cure. Next. Proceed. This particular thing increases actually the scientific challenges, and in this particular aspect, when we look into next, it actually increases the object of the drug discovery is to increase the safety, delivery convenience, individualized treatment coupled to diagnosis, unmet needs. As you know, that particular new disease COVID-19 has emerged. We have to find out a solution to it, and the drug actually, the new drug must be affordable to all the people. Next. So in this, whenever we talk about this particular process itself can take 1 to 2 years. That after that thing, once the approval comes, post-market surveillance can also be seen. This is a long drawn and cost prohibitive process. So in this particular case, people are trying actually the digital technologies, but when you are talking about computer-aided drug design, we can actually decrease the discovery phase by virtual screening. As the other day, Dr. Sastry was mentioning the chemical space, 10 to the power 181, the compounds which are available, and people thought originally it can be screened all using high throughput screening, but that is not actually feasible even after having actually the ultra screening aspects. So now we are actually going into all the training what you are getting is virtual screening of the data banks, whatever actually the data sets which are available. From there, this can be reduced. Next slide, please. So in this particular case, next one. Now, the basic concept of actually the drug receptor, when it comes, the drug has to interact with the macromolecule, and then only it can produce actually the aspect for making actually new drug discovery as well as actually in cases of therapeutic design. Next, please. In this particular case, when we look into it, the drug receptor interactions, the original concept was given by the Langley as early as in 1875, receptive substance. the initiation of the response involves the formation of a complex between the drug and the active site, so that actually the reaction will take place and an effect is produced. Initially, receptor theories were proposed and the neurotransmitter substance, the body was actually determined. Paul Ehrlich was the person who actually talked about unless the ligands or the drug interact with the macromolecule, the effect is not produced. And specificity and magic bullet answers were actually the more words which was used by Paul Ehrlich. Later on, when we talk about it, the drug receptor interaction concept was furthered by Clark, Arians, and the Stevenson and Perchgott. Next, please. This particular aspect, when we have to see, in this actually the receptor can be present on actually the plasma membrane of the cell surface, and it can also be present intracellularly in the cytosolic receptors within the cell. Now, why do the receptors are present in the plasma membrane? Because of the physicochemical properties of the substances, they may not cross the plasma membrane. So hence actually the receptors are present on the plasma membrane for actually the sending the communication, because this interaction or drug receptor interactions leads to communication and messenger systems. So in that particular case, the substances which can cross, something like steroids and other actually the hormones can cross the membrane, which are actually having the optimum the physicochemical properties, they can actually act on the cytosolic receptors. Now, once actually the receptor interaction takes place, agonist, whatever we call, which produces an effect, and it interacts with the receptor, a complex is formed. That complex is in the active state, the receptor, which generates actually the stimulus. In the reaction, whatever it is shown, next we have actually indicated a double arrow indicating that several steps are involved. And where actually initially it was talked about black box, nowadays the various actually the processes which are taking place within this particular black box are actually the transmembrane signal mechanisms are actually deciphering. That results in the stimulus response. The magnitude of the stimulus response depends upon the two parameters. One is the concentration of the agonist which is present at the receptor site. So directly proportional to the concentration up to reaching the saturation point. And it also depends upon how much actually the signal is developed because of the amplification. So in that particular case, these are the two determine stimulus response aspect. Next one, please. Hello, next one. Next slide, please. See the various actually the receptors which are actually the present site of action are given in this particular actually the slide. G protein coupled receptors which are actually having the rhodopsin family, actually adrenergic, angiotensin and muscarinic and serotonergic receptors. And you have a next category when we talk about enzyme associated receptors and ion channels, where actually the channels are mediated and GABA A and glycine and 5-HT3, and voltage dependent are gated channels, sodium, calcium and potassium ion transporters. Then you have actually the transcription regulators and the steroids, which are actually coming into the factor. And some of the things which act also on actually not through specific receptors, examples are given. Next one, please. Hello, next one. Now, I'm mainly concentrating on the G protein coupled receptors. This is the structure of the G protein coupled receptors, plus actually the next one again, which shows animation. Okay. Now it is a 7 actually the transmembrane receptors across the plasma membrane, the proteinous in nature, and it is actually coupled with the G protein, as the name indicates, The G15 is a heterotrimeric compound, and in that particular case, G alpha, G beta, and G gamma. But functionally, it is a dimeric, that is G alpha and actually G beta gamma. And when actually ligand interacts with the cell surface on the cell outside of the cell surface, in that particular case, the initiation of this thing takes place, and the effect actually generated into this particular case. So the effector, it actually controls the effector, and stimulation of the effector takes place. That leads to signal mechanism. This is how actually the signal, whenever a compound is interacting, ligand is interacting outside the membrane, it is actually the producing effect inside the cell, and actually the physiological effect, whether it is a contraction or release of secretion, whatever the things it can happen. Next one, please. Go to the next. So G protein coupled receptors interacts with actually orthosteric ligands as well as the allostatic ligands, which can be agonists, which are actually having affinity as well as actually ability to induce the response. Agonist, antagonist is having affinity towards receptors, but it is not having any capacity to induce actually the response. So allostatic, actually other than the site which actually they interacts, this can be full agonist and partial agonist. How actually the classification from 1995 before and after 1995, how the system has done is actually depicted in this picture. Next one, please. Hello, next one. Next. This is actually the quantitatively, when mainly the drug receptors can be characterized by pharmacological responses, that means end organ responses, and that can be quantified, and quantitative pharmacology, we can talk about it, and the representing these particular curves. This is against actually the alpha-adrenergic receptors mediated responses in an isolated rabbit thoracic aorta system, where actually the dose response curves are towards actually right, means they are having the decreased affinity. That means it requires higher concentration to combine with the receptor and produce the stimulus. And you can see all actually the series of the compound phenylephrine, amidophrine, and cerazolin, and actually ST587 and SGD, they are actually the decreasing variable affinity, variable ability to induce the response. A response can be treated at 100%, whatever in a particular system, and that actually in the same system, it actually having decrease in the maximal response. Even after increasing the concentration, it cannot achieve. So these actually decreased ones are called partial agonists. The next one, please. This actually, next one clearly indicates, now the receptor number in the system is constant. That is what we call Vmax, that I will explain it actually in the later slide. And is all the receptors are required to occupy the produce the physiological response, that is in this particular case a contraction. Pharmacological response which is mediated is a contraction. So in this, what I want to actually convey, the 50% of the response whenever we are looking at it is produced with occupation of this on the x-axis, receptor occupation. This studies can be done by using the receptor occlusion studies. That means a receptor can actually, the compound, which are alkylating agents, can interact with the receptors, and that is converted into alkylated receptors that is no longer actually equivalent to original receptor. So with that receptor occlusion studies, one can actually determine the percentage of the occupation and the response which is produced. What I want to convey in this picture is 5% of the receptors are sufficient enough to actually produce a response, a 50% response. A non-linear relationship is existing. On the other hand, the partial agonist, whatever we are seeing, it has to occupy 100% of the receptors, even then it cannot get it. So that clearly indicates that depending upon the efficacy, this word actually used by Dr. Vartan, varies from compound to compound. and its ability to induce the stimulus generation. So in that particular case, there are actually certain compounds which actually have lesser activity, lesser efficacy in producing, and thereby the response will come into the picture. Now the importance of the spare receptors comes into picture. These actually 20% of the receptors are spare. In some of the cases, it will be around 90% of can be spare receptors. Neurotransmitter inside the body, when you study it, norepinephrine in rabbit aorta do have actually 90% of the spare receptors existing in it. So in that case, when actually the age progresses, the downregulation of the receptors can occur, and a partial agonist response will be completely less or even antagonist. It works as an antagonist, but a full agonist still can produce in fewer receptors. Next, please. So, hello, next one. The receptors further can be characterized by using actually the receptor binding assays. And here in this particular slide, what is being conveyed here is a radio endpoint is determined radioligand binding assays. So in this particular case, the iodinated compound, which is used angiotensin 2 in producing actually binding this one, and you can see receptors are saturable, and it actually reaches actually asymptote, and this particular curve, the nonlinear curve, is converted into scattered plot, and from there actually the Bmax and actually the Kd value can be determined. Kd value is the concentration which is required to occupy 50% of the maximal receptors, Bmax. So that can be done and find out actually the Bmax as well as actually the affinity of the compound to the receptor can be determined. In the second panel, what I want to convey is in the B panel is the competitive binding assays can be done. Again here, the prazosin tritium labeled is used compound, and in this particular case, a test compound, and actually test compound is actually the solid line, and whereas the standard compound or reference compound is against 1 is in the dotted lines. From this curve and IC50 values, you can understand the standard compound is 10 times more potent, or in other words, the test compound is 10 times less potent on the IC50 value. That means inhibitory concentration, which is required to produce 50% of the inhibition of the binding. So the next one, can I go to the next one, please? Now, hello, next one. Now, as I mentioned in the beginning, agonists we have talked about it. We also have the antagonists. Antagonists, they bind to the receptor, but they cannot produce it. Any actually measurable response. In that particular case, we have to use actually agonist, and how that dose response curve is actually shifted without suppressing the maxima. In all these cases, yohimbine is used an alpha adrenergic blocker, which is actually shifting the dose response curve towards actually the right without suppressing the maxima. In all the cases, the maxima is maintained and shifted towards actually right. All are parallel in actually the nature. This is called actually the dose ratio. From the dose ratio, one can actually determine shield plot, Arnold Lakshana shield plots, and pH value. The affinity of the antagonist can be determined with actually Arnold Lakshana shield plots calculation, that is pH scale. From there, we can understand pharmacologically which one is having affinity towards a particular receptor. That is actually here yohimbine. It's the pH value comes around 5.97. So the second panel, when we are talking, you have to understand actually the partial agonist. Partial agonist it not only acts as agonistic and produce the response, and it also actually works as an antagonist. That is what exactly theoretically shown in this particular picture here in that second panel, indicating that up to the ability, the intrinsic activity of the partial agonist, this is a particular synergistic activity, agonism is applied, and after that particular point, this particular thing is showing the antagonism shifting rightward actually the case. So that is actually the nature of actually isolated actually the preparations pharmacological issues and understanding responses which are produced by the agonist and how an antagonist interacts and a partial agonist interacts with it in presence of it, those response curves. Hello, next slide, please. Next slide. Now, the other day, Dr. Sastry was talking about the drug discovery and biology and mathematics in his lecture. He mentioned there are so many variables. Some of the variables, why the response are changing. So now the binding to G protein coupled receptors, it also actually mediates not only through G proteins, but also through beta arrestin. So there are actually one is actually the G protein pathway and another is beta arrestin pathway. So in this particular case, depending upon the ability to which of this pathway is activated, you will get actually the effects. If it is a non-selective, then in that particular case, simultaneously that particular substance will produce both actually the effects. So in that particular case, what we want is in the multiple signals when they are mediated, we want a preferred activation of one specific single pathway over the other by the ligand kinetics. So if that happens, you will get a distinct desirable effect. So you can eliminate the side effects. This is what is called biased agonist or biased agonism. This concept is now very much coming up and still actually in the case. Next slide, please. Hello, next slide. Now, to understand this a little more, the possibilities of actually as I said, the trimeric form, G alpha, G beta, and G gamma, in the body when up till now identified 16 G alpha and 5 G beta and 13 G gamma subunits combine and form wide range of heterotrimeric G proteins. So that is one of the major actually fundamental points. So now in addition to it, you are also having actually beta arrestin. So in that particular case, how do we actually get a differential activation? We have to see. That is what in the next slide. So next slide, please. Hello. Okay. Now this actually I will take a little time to explain to you. Now there are actually 3 receptors. Imagine imagine one hormone is acting on R1 and R2 receptor, and there are 4 actually the G protein are present, G1, G2, and G3 and G4, which are coming into the picture. So in that particular case, when you look at this particular thing, the network is actually the formed. And what is that effect? You will get actually it can actually go to the 3 effectors, and 3 types of effects can actually come into the picture. This is more of actually the neural networking. When you are actually working on the programming in the neural networking, you can actually find out what is the best way of actually compound and to get actually desired actually the action over actually, because that clearly indicates any drug is having a primary action for which actually it is prescribed, and simultaneously the same drug can have also what you call secondary effects or secondary pharmacology. One is the primary pharmacology and the second one is the secondary pharmacology. That's why the concept of actually the polypharmacology started. So in that case, the drugs can have the different effects because of the side effects and polypharmacology. Some of the drugs which are introduced has to actually withdraw because of the toxic effects it also produces. So in that particular case, how to actually get that particular desired activity is one of the major points. Next, please. Hello, next, please. This is biased agonist. When we are talking about it, say now there are two pathways as I mentioned here previously. One is actually the G protein and G alpha I, G alpha S, G alpha Q. That is the major ones. Though the 15 are there in that particular case, and beta arrestin and beta arrestin 1 response, beta arrestin 2 response, and overlapping responses can actually take places. Now, so in that particular case, a biased agonist, which actually works on where and which pathway is more important. As per actually one of the examples, what I want to convey is voliceridine, which is actually an analgesic. When you activate actually with this compound TRV130, it activates the beta arrestin pathway and produces analgesia, but it do not produce actually the side effects. This is already in the phase 2 clinical trials, phase 2B already, this particular thing. And in that particular case, this particular concept, how to get actually the selective stimulation of one particular pathway is actually the next concept. The next slide also, in a different way, it is explained the same phenomena. This is what exactly typical agonist, both actually it produces a side effect and actually the therapeutic effect when actually they cannot distinguish the two pathways of the G protein coupled receptors. Both actually it will have a side effect as well as actually therapeutic effect. On the other hand, if it is a functionally selective ligand, it is functionally selective ligand, what I'm using. So in that particular case, or previously the scientists have used, so this actually produces a therapeutic effect, but not actually the, what you call, the side effects. So in that particular case, we get actually the aspects. So next one in that particular case, when we are talking about, hello, next one. So the explanation are the performance of drug may change in different systems and the tissues and person to person. So in that particular case, the density of the receptors is one factor. Basal actually the activity, how they are activated, constitutionally activated in certain actually the conditions, and receptor effector coupling efficiency, how good they are actually coupled, and how efficiently they can convert actually all this thing into signal, and downstream pathways. As I mentioned, there are several downstream pathways which are actually there. So in that particular case, which pathway the compound is activating selectively or non-selectively activating, how the effects will come, and the receptor activation responses and the ligand and the spare receptors existing. And in addition to it, the disease conditions, age, chronic administration of the drug, because chronic administration of the drug can upregulate. If it is an antagonist, it upregulates the receptors. And if it is an agonist, it downregulates receptors. We have not talked about the receptor regulation. And in the disease conditions, a very simple aspect when we talk about achlorhydria. So naturally, the pH actually in that stomach area, it will be less. Achlorhydria means there is no hydrochloric acid secretion. Compounds, you know about it, chemicals, which are actually follow the Henderson-Hesselbach equation, and it is only ionized compound which crosses actually the membrane. So if the hydrochloric acid is not released, the acidic drugs from the gastric actually, that is stomach, it cannot be absorbed. May I have the next slide, please? Hello, next slide. Now, after actually getting the aspects about the concept of the discovery and pharmacological, that means once actually the lead or hit and determination is done, you have to prove the assay biological screening, what we talk about it, that particular aspect is actually HTS, automated biochemical or cellular assay, or in vitro, actually the assay, biochemical and secondary assay, cellular actually assays can be done in vitro stuff all food receptors. So whereas it's a non-selective means it is equally effective at alpha 1 and alpha 2. So this is one actually fundamental point we have to understand. And then actually ADME, that it can be predicted in silico, and in actually the in silico results needs to be validated by using the experiments, by permeability studies, and actually the protein, actually the binding assays, and actually what you are talking about, cytochrome P450 needs to be done. On the other hand, PK profiling needs to be done. PK means for pharmacokinetic, ascending dose, single ascending dose, and multiple ascending doses, and mechanistic actually the studies, tissue distribution. One of the how much actually the various compartments it is going into the base physiologically based pharmacokinetics, what we talk about it, and the PD models, PD studies one has to do. That means pharmacodynamics. That's what I explained, the primary targets, and we have to determine PK/PD and mode of interaction and PK/PD correlation, and the physiologically based pharmacokinetics is another aspect which needs to be understood. And on the during this period, we have to do actually toxicity studies. Toxicity studies, pilot toxicity studies can be done in a non-GLP laboratory, and the final results are required under actually good laboratory practices for regulatory approval. So these are actually the outcomes of this one at the bottom, which is actually the mentioned at the bottom of it. May I have the next slide, please? Okay. So this is again what various actually optimization process, how the various parameters are determined are actually they mentioned, which is there may be some repetition in the previous slide. So in that particular case, lead optimization, so potent and efficacious assay, and I will talk about the third point, lead optimization, and specificity and selectivity, increased specificity is required, and we have to do optimal ADMET tests and PK profiling, and the metabolism. One of the major things actually the drug-drug interactions do come because of the CYP inhibitions, cytochrome P450 inhibitions are actually even actually this particular thing one needs to be identified, so that the simultaneous administration of two categories of the drugs needs to be the candidate which needs to go negativity in HERG, that is actually the increase in arrhythmias, one has to actually look into the case, and PK parameters needs to be identified, and it must have a wide margin of safety. That means wide margin of the safety means the therapeutic value in the 50, that means ED50 value, effective dose at 50% of the case versus actually lethal dose 50%. It must be a wide actually the range, that is actually therapeutic window is solved. The more the window, the drug is more actually the safe in this particular case. So next place, when we talk about after this thing, this is what exactly I was talking about it, PK, that means physiologically based modeling of the pharmacokinetics. We have to get actually in vivo, in vitro extrapolation situation, and the system data. The lot of actually the computational work can be done, and in developing actually the PB and PK modeling and structural model based upon these actually compounds, the studies components, you will get actually study design and drug administration can be done in this particular case. One, this is what actually, because a series of actually the toxicity testing, starting from acute toxicity, means a single dose needs to be administered and find out the case, how much actually the dermal toxicity, dermal irritation, eye irritation, skin sensitization, and other actually the aspects. And subchronic means it is a what you call multiple administrations for 90 days. The same actually the procedures and how much actually the survival rate and any of the behavioral aspects needs to be gross behavioral aspects, and it needs to be done at the end of the study, the sacrifice animal and find out histopathological differences, anything are present in the treatment doses. So the chronic toxicity is much longer than actually depending upon the nature of the category of the drug substance, whether it is actually long-term, lifelong, the drug needs to be administered, chronic toxicity studies needs to be done. And the next category is carcinogenicity, whether actually they are producing a cancerous in the body, cancer or tumor growths are coming into the case. One needs to actually find out that particular thing to avoid that will not, the drug may not cause actually the case. Reproductive and developmental toxicity has become a mandatory nowadays because of the, you all know about the thalidomide incidents in early 1960s and phocomelia development. In that particular case, the babies which were born are concealed limbs. So the reproductive toxicology and developmental toxicity testing is actually introduced very much actually that is an essential. And the next one is genotoxicity. That means how the drug interacts with actually the DNA. Is there any interaction? And it produces actually the toxicity by studying Ames test or in vitro chromosomal aberration. And then actually the micronucleus test one can actually carry out. Next please, next slide. Hello, next slide. Now the clinical trials, after having this particular thing, one needs to actually apply and with this data investigational new drug application for the US FDA is actually referred it, and European Union also the same, and the different countries do have nowadays ICH guidelines have come, International Committee on Harmonization, so that if multi-country marketing, if one wants it, ICH guidelines can be followed. Phase 1, briefly to talk about the phase 1, it is in the healthy human volunteers, is a less number, usually 20 to 80 people it is conducted, and regarding the safety and tolerability of the new drug needs to be determined. And phase 2 is the efficacy and as well as the side effects in the patients. Enroll the patients, that is again a limited number, 100 to 300 people, and actually they generate efficacy data and the safety data. And at every stage, the drug authorities needs to be given approval, giving the green signal for going to the next stage. And phase 3, actually the trials, when we talk about it is multi, that is actually multi-centric and global trials. Nowadays they are actually large global trials, different countries, so that all ethnicity people will be taken into consideration, and the numbers can go up as much as high as 20,000, even more, to prove the safety and efficacy. Can I get the next slide, please? This is what exactly what we talk about the clinical trials. And Indian actually, Dr. Shastri asked me to talk also Indian. This is, I am not going into cover because of the time schedule. These are the details are given at this particular slide. One can actually reach and find out. A majority of the points I have covered it, and Indian guidelines can be seen, and the application how to file it in this particular aspect is given. Next slide, please. This is where actually my first part of the lecture is over. That is actually the drug discovery and development. And the main actually the topic which is coming is the what you call COVID. You all know about it. The bat is actually the source of the virus. An intermediate source, we already have the previous actually the coronavirus, which is actually SARS-CoV-203 initially, and that is a very limited. And then actually the MERS, the COVID, that is in 2012, a limited number. And today, this is a pandemic is already declared and running into 26 million throughout the world, and somewhere actually 8,66,000 people more than are actually the succumb to the disease. And India, when we talk about already, we have reached actually 3 million and ready to touch 4 million, and somewhere around 68,000 people are dead. The point what I want to convey is that is actually reservoir of the so many viruses. So still it is not able to get the infection very much. The normal actually the bat, whatever we are talking about it. So in that particular case, one has to understand why actually the bat is not getting infected and actually the severe acute respiratory syndrome is not occurring. It gives actually the clue. This is called actually the comparative studies, evolutionary point of view one has to study, and then only we can understand where actually human immune system is different from actually the bat system and how actually so much havoc has created in humans. Can I have the next slide, please? This particular thing. The interesting aspect when we see this pandemic, the number of actually the papers which have already actually appeared up to May, somewhere around on the month of May alone, 8,000 papers, and more than actually somewhere around actually 20,000 papers have come across already up till now, up to the end of the May point. So in that particular case, because of actually the throughout all over the world, this particular pandemic has occurred. Can I have the next slide? So much actually generated in the scientists. In general, when we talk about this thing, it is an enveloped virus and single-stranded RNA, and mainly when we talk about actually 4 types of genetic actually the aspects, 4 types of genera are present. Alpha coronavirus and beta coronavirus is now responsible for this one coronavirus, and gamma coronavirus and delta coronavirus. Next, please. Hello, next, please. So in this case, the main aspect which is coming is causing the respiratory and intestinal infection. The latest reports indicates that actually in the gut microbiota and the brain and lung axis is working in this particular case. It is absorbed from the gastrointestinal tract also and causes gastric disturbances. Can I have the next slide, please? Hello, next slide. This is what is the pathology, mainly when we talk about acute cardiac injury, one of the major actually the thing, multiple organ failure, and acute respiratory disease syndrome, cytokine storm. These are all sequel to this thing. Nowadays, people are talking about the bradykinin storm also, and we have to understand how actually to develop a drug substance, what are the things which are there essential to understand a little bit of pathology and pathophysiology of this particular disease. Can I have the next slide, please? This is how actually attack from the source, the coronavirus SARS-CoV-2, which is scientifically called, and this particular actually the virus, which is actually spherical and actually having the spike proteins, and through the angiotensin converting enzyme 2. That is my interest. Why actually I'm not a virologist, I'm not in infection diseases, but the angiotensin converting enzyme, because it is a G protein, and actually the angiotensin converting enzyme 1 actually the mainly plays other role in hypertension. This angiotensin 2 is actually the receptor for the virus to get into the system, and it is endocytosis and receptor mediated in that particular case. And this actually acute right at this moment, the death mainly due to because of nowadays brain and the lung axis. Otherwise, it is severe actually respiratory syndrome, and the comorbidity is in the old age, chronic actually pulmonary diseases and cardiovascular disease and diabetes. These are all old age phenomena. In those actually the conditions, the patient if is infected is going to have actually the more severe attack, and one needs to take actually the precautions. Next one, please. Hello, next one. So even though 17 years back, the first SARS has actually come into picture, there is no drug. In fact, in 2015, Gates actually pointed out that the next actually world catastrophe may not be nuclear war, but it is due to microbes. So in that particular case, nobody has cared about it, but we have no actual therapeutics. However, drug repurposing, already someone actually talked about drug repurposing, and is the only solution to get immediate answers. There are actually the certain things which, say, for example, corticosteroids are playing a good, that is one of the main thing, decreasing the severity. Next one, please. Hello, next one, hello. What actually I think it is somewhere around 26,000 to 30,000 bases, which are actually the largest genome RNA. You are having actually this virus is having three membrane. That means a spike protein and envelope protein and M membrane and nucleocapsid protein. And these are actually the things which are essential proteins which are existing in the virus, which actually play the role. S protein is mainly used. Spike protein is to bind the receptor and get actually viral entry into the host cell, and then M and E play actually important role in viral assembly, and N protein is necessary for RNA synthesis. So next one, please. Hello, next one. Next slide, please. This is actually what the structure which we can actually talk about it. We have explained it. It is having the spike. Corona means actually, corona means it is a crown-like, as the spikes actually called is a crown-like structure. So in this particular case, the membrane glycoprotein and then spike protein, and actually how the spike protein combines with actually. And the mechanism is actually shown in the next slides. Okay. Hello, next slide, please. Next slide. One of the major important thing is ACE2 is expressed throughout the body from the top to bottom. ACE2 that clearly indicates that the virus can actually enter in the different organs. One of the most important aspects. So starting from the nasal cavity to the tongue and then actually the lungs, you can actually talk about heart. In fact, there is a common language, what we call head to toe. Starting from the head to toe, every organ which is actually having the expression of ACE2, that means the virus can actually invade these different organs. That is the reason when we talk is starting from anosmia, that means no smell, actually the first actually the physiological or symptom. When we say agousia, which is called actually the lack of taste buds are actually affected, and even eye through the eye, and that's why they say whole the face needs to be covered with actually the guard, and starting from everything, this particular actually the thing is coming into the picture. But one interesting aspect is the death is more in actually the males when compared to females. The androgens are playing, there are several reports indicating that androgens are playing a role in actually severity of the disease, whereas androgens are not there in that particular case. The females, the less actually the activity which is coming. Recent actually the reports are coming out. It is also present in the placenta. The ACE2 receptors are also present. Still actually the indications are there. There may be some effect on actually the pregnancy. Also the reports are actually coming out. I may have the next slide, please. Hello, next slide, please. Now, my interest when I said actually why I'm actually talking about the coronavirus is angiotensin and actually the ACE, angiotensin converting enzyme, because in the hypertension it plays a role, and in diabetes and in actually the hypertension, this particular ROS, renin-angiotensin system, plays a role, and angiotensin converting enzyme is prepared in the lung, and the inhibitors, which you all know about, is ramipril and other categories of enalapril. All these actually the substances inhibit actually the case, and it is used in hypertension. This converts angiotensin 1 into angiotensin 2. Angiotensin 1 is a decapeptide. From there actually it is converted into octapeptide. So the angiotensin 2 acts on two types of subreceptors, 81 and 82 receptors, and ACE2 cleaves angiotensin 1 to angiotensin 1 to 9. That means it is actually removing one peptide, and angiotensin 2 to angiotensin 1 to 7 peptides. That means amino acids. So this angiotensin 1 to 7 actually acts on 82 receptors as well as the MAS and the MARG-D receptors. And that is what is very important. It produces this particular angiotensin 1 to 7 produces opposite effects of actually the angiotensin 2, what it is producing. Next slide, please. That is what exactly it is explained in the next slide. Hello, next slide, please. So the angiotensin 2, when it is combines with angiotensin 1 receptors, vasoconstriction, inflammation, proliferation, and cardiac hypertrophy. That is not desirable. That's why ARBs, angiotensin receptor blockers, which are actually used, that is losartan, and actually the series of compounds, actually sartans are used. Telmisartan is also having antidiabetic activity also. Now, this particular actually the other axis, angiotensin 2, that is forming actually angiotensin 7 by the ACE2, it is natriuretic and cardioprotective, anti-proliferative, and endothelial protection and vasodilatory. It is quite opposite in these two axes. So that means it counterbalances the particular thing. So now, if ACE2 are actually ACE2, which uses actually the next place, ACE2 combines with virus, and it actually internalizes. So when it is internalizes, ACE2 also goes actually into the cell. So there is actually down regulation. People thought actually that particular thing. So it should be actually the contradictory, the negative, the positive aspects of ACE2 mediated effects are lost, and in actually the already who are taking actually the ARB, that means angiotensin receptor blockers, sartans, need to be discontinued. A particular argument came in that case, but that is found to be not correct argument. There are several associations who are eminent bodies controlling the regulation and diabetes. As said, there is no necessity of discontinuation. One of the positive aspects of that one is actually because the next slide actually shows the point to be there. It upregulates the receptors in that particular case whenever. Now, once actually the virus gone inside, that is actually with the help of TMPRSS, the transmembrane series 2 will actually split this particular thing, and along with actually the spike protein and receptor binding protein, it goes inside the cell and form the endosome. That is actually the first step, and second step is fusion with endosome and goes inside. That is the third step is from the endosome, it is converted actually the proteolysis takes place. Whatever it is in the endozyme, the lysosomal action, that actually the uncoated actually the RNA, single stranded RNA released into the cytoplasm. Now this actually single stranded positive RNA is because of the replication complex, and it is actually formation of the negative RNA strand and actually the positive RNA strand, and then the replication process actually takes place, and then actually the assembly, and again actually the new virus is synthesized in the Golgi body and complete actually. That is how the replication takes place of the virus inside the cell, and that the replicated actually the virus, which is by exocytosis, getting out of this particular actually the system. That is what which actually happens in this particular case. So the next slide gives the mechanisms in this particular case. So when we talk about it, the first step, can we actually inhibit TMP-RSS2, or actually other actually the proteases, which is also protein? The TMP-RSS2 is inhibited by chemostat, which is already chemostat is approved in Japan for the pancreatitis, and that can inhibit, and that actually the control presently, this is in the clinical trials right at this moment for actually getting the approval. The most important aspect is when actually the endosome, and the particular thing, it requires actually the acidic pH and for the proteolysis, but whereas actually the hydroxychloroquine and the chloroquine, more correctly, both of them are actually, hydroxychloroquine is less toxic when compared to chloroquine, which increases actually the pH of the endosome. That is one of the major actually the point, and thereby the replication do not actually takes place. Unfortunately, people actually talk about its previous slide, previous one. Hello, previous one. It proves that actually the pH increase is actually preventing it, but it is also reported to have an arrhythmic property. That is one aspect which actually taken into case. And RNA dependent polymerization, when we talk about it, it can be inhibited by lopinavir. And we have actually other substances, and whatever actually the symptoms which are coming, though the virus is not affected, but at the same time, lungs which are actually through macrophages, the IL-6 and TNF alpha, which are being actually released, that particular case to be inhibited by tocilizumab, that is actually monoclonal antibody, which can actually prevent it. Now, the recent reports after the slide preparation, there is one more actually the point which has come is corticosteroids, which can also inhibit actually this particular that is immunomodulators activity is there. So we are at this particular stage. So in that particular case, what are the sites which actually one can actually think about it in doing actually the case needs to come. Next slide, please. May I have the next slide? Now, how to, once you actually get the compounds, whatever actually the proteins, whatever you have used, are going to select and use it, and the binding studies, we have to have actually validation of those actually compounds, whatever the bindings which are there need to be done in an experimental model. Now, these are the various species which are laboratory animals which are used, chick and duck and pig. There is no actually expression or actually the infection of the coronavirus in these species. However, guinea pig and ferret, dog, cat, and common marmoset, rhesus monkeys, cynomolgus monkey, and African green monkey, and human beings, there is actually the severity of the infection which is available. This is phylogenetic actually the case. One can actually talk about it. These are the models can be used. People have actually used mice. Mice is also, yes, I have mentioned mice also is there. Mouse, okay, in that particular case. May I have the next slide, please? Now, the next slide, yes. the mice models and the SARS-CoV-2, the common actually the animals are actually used in the skin bulgy variety and C51BL6. At the same time, there are actually the deficient mice also, that means knockout mice. One of the latest actually things which are being used nowadays, latest means for last 10 years, using actually the other actually the SARS virus, which are being done. And in this particular case, human S2 are expressed in actually K18 human S2 mouse model. So this actually the mouse model, which is a transgenic mice, and having actually the mouse S2 receptors is also expressed in them, as well as the human actually S2 receptors, and cytokeratin 18 is used as actually the promoter. In this mouse, it actually expressed in the lungs, colon, and moderate expression of the S2 receptors in actually the case human actually S2 receptors. May I have the next slide, please? Hello, next slide. Now, when you see what are the symptoms which are coming into the human clinical features are also expressed in human S2 expressed actually the mouse model. High viral actually the lung titer. So that means one of the major important aspect where the site of action and the death is coming because of that thing. So pro-inflammatory cytokine production. So that is both in the cases it is observed. Lung damage and pulmonary decline. One of the lung damage is irreversible, and once the disease is actually the complete recovered, still actually the lung damage is there. Permanent damage, ground glass phenomena, whatever they talk about it, and clinical signs, weight loss and pneumonia is occurring both the cases. This is what is having actually the similarities between the two things. May I have the next slide, please? Next slide. So this particular thing, when we talk about it, these are the animal models. How do we actually translate it completely into this particular thing, a new drug, once ever this thing called? This is called translational studies. One needs to understand the animals are animals. They have certain similarities. At the same time, they also have dissimilarities. And so in that particular case, we have to prove that particular thing in the clinical trials. That is what is the whole story, what we are talking about. It from the cell cultures, directly in the in vitro cell cultures, one can actually talk about it. Now, from the cell culture, actually the new phase which has come is organoid. That is nothing but actually 3D cell culture, where actually it takes the shape of an organ and the cell culture automatically. And from there, we have actually the animal, actually the aspects. We can go into that particular thing. And from the animal studies which are successful, one can actually go into clinical trials. It is a long drawn process. Okay. Okay. This is what. This is the next. This slide actually talking about what are the proteins which are actually what I talked about it. These proteins may be virus protein, that is 3-CLPO and PL3O, that is like RDRP, and that is the S. Next, actually S protein interaction and helicases, as well as actually nuclear NTPases, and the human TMPRSS and furin. Now, as per the latest reports, which has appeared in 30th August in this journal, somewhere around 12,000 compounds have been tested, and actually the results are available against this particular thing in the cell cultures and animal bioassay. This is called bioassays, not in silico. Bioassays, whatever actually the compounds are. You can understand in 8 months how many compounds have been screened, coming from the virtual screening aspects, how actually the progress has been done is actually coming into the picture. So the next slide, what I thought somehow Okay, next slide is indicating these actually the how many documents which are actually coming on each topic, which is also this paper actually gives, and number of the substances on each paper, how many actually things on the various actually sites which are present in the COVID. Whoever is going to plan to work and can actually take this particular as a clue, and one can actually this paper, please refer to this paper and find out, and your actually the studies and your virtual screening pattern, you can decide and go ahead and do a particular aspects in this case. So I actually put one animation that is actually the transmembrane signal mechanisms. Okay, automatically this is what angiotensin receptor, how actually the this thing effects will come into picture. So the angiotensin when combines with AT1 receptor, G alpha and G beta, and G alpha will activate actually phospholipase C and converting actually PIP2 and into IP3. Okay, IP3, and IP3 activates on IP3 receptors, that is intercellular calcium is released, and whereas actually other part actually PIP2, IP3 plus diacylglycerol is actually releases, activates PKC as well as actually the L type of calcium channels, thereby the calcium actually comes into picture and the contraction comes into aspect. There is a functional coupling. That is why my interest in angiotensin, and similarly AS2 is coming into the picture, and there actually 82 receptors is causes actually the relaxation. This is what. So to conclude finally, the next slide, please. I kept it last because I thought I can present it from my side, whether somehow the system did not work. I'm sorry for that. The conclusion of my talk, and we bring about it that one, that drug discovery and development is a time-consuming, cost-prohibitive, multidisciplinary, and these multidisciplinary are supplementary. They are not independent. They have to cohesion. They have to work in cohesion, and communication must be there in different actually subsections of the drug discovery people. Then only it will be. It is actually a knowledge intensive, knowledge-based process. Computational science, computer-aided drug discovery, and digital technology is changing the scenario of R&D and development in the field of diagnostics and therapeutics. Yes, SARS-CoV-2 is a game changer. People use it for another thing. I'm not quoting that one now. And it is invigorated interest in the drug discovery and development. I hope India can actually succeed and become Atmanirbhar in producing some drug in near future. Thank you very much. Thank you very much.
Thank you, sir. Thank you for your wonderful lecture. Now I request Dr. Himakshi Sharma to moderate the question and answer section.
Sir, am I audible, sir?
Yeah, sure.
Thank you, sir, for your very informative and conceptual lecture, where you have mentioned from the cellular side of drug actions, then you have given the example of a very important receptor, and then you went to the pharmacodynamics, pharmacokinetics with the example of antagonist, agonist, and then you explain about the in vitro assays, which are very important, then that need to be carried out before a particular drug out to the market. And then most importantly, you have shown a very vast important information regarding the SARS. Within this short period of time, sir, you have very important points of drug discovery, and then related to SARS, what are the recent experiments are going on. So thank you very much, sir, and it's our privilege to have you in DDH training program. And we received some questions. So because of time constraint, I will take maybe 2 to 3 questions, sir. So shall we start the question answer session, sir?
Yeah, thank you very much for your comments. Okay.
Thanks. So, sir, first question is, targeting a receptor or enzyme may lead to damage of normal cellular machinery. So, how to deal with this while we are designing a drug for a disease?
See, we are targeting means xenotic, what you are trying to talk about it. Whenever actually there is a necessity, sometimes we use the neurotransmitters itself, norepinephrine in hypotension, we use norepinephrine, adrenaline, also in certain cases. When endogenous substances are actually used as a drug, it is not going to harm many of the biologicals now, whatever we are talking about it, certainly it will not produce any harm. But only the question comes is persistent increase or persistent use, that means chronic administration, can downregulate the receptors. On the other hand, when you are asked about the drug substances which are genotoxics, which are not actually available in the body, if they are used, if they are not safe, certainly that is all actually all about the toxicity studies which we have to do, and we have to find out that is the reason nobody wants to directly start a new drug on human beings. Number one, nobody is volunteered to test a new drug without proving its safety and efficacy. So in that particular case, first we study the animal aspects and find out any of the adverse effects which are there that needs to be. That's why I talked about actually in my slides, so that is bias recognition and why actually these toxic effects are coming. So we have to find out actually a substances which we have the selective activity and specific activity, rather than it is having so many secondary pharmacological activities. And one aspect which we have to understand, people should misuse certain words. One is a side effect, another is adverse effect and adverse event. So these two are the needs to be differentiated. Side effects are tolerable effects, and you get actually when you talk about the risk-benefit analysis, any particular thing when we are talking about it, risk-benefit analysis, it should be favorable. More benefits should be there. If a drug substance actually causes a cure for a particular disease, it causes nausea or a vomiting, slight actually nausea sensation, yes, one can tolerate it. But if it produces actually early organ, that is not desirable. So one need not actually go into that particular thing. So this is what whole actually the aspect of toxicology are actually the toxic effects and finding out the responses. Even in the post-market surveillance also, if the different genomic population, genetic populations are there, ethnic populations are there, any adverse effects comes, the drug is recalled. It's called pharmacovigilance, which I have not covered. Thank you.
Thank you, sir, for your well explanation. So, second question is, if the receptor active site is mutated and it is no longer recognized by a particular drug, can it will be possible to inhibit that particular receptor by targeting the allosteric site?
You shared something, but I can't see the screen.
Mutation means, see, there are 2 points what you want to convey the message. It is a very good question. Normally, what we say, if it is allosteric site, still it is functional, it can be used. Whether allosteric site also it is functional or not, mutation occurred or not, your question will not say. There is a possibility when the mutation occurs, allosteric site also will be mutated. The possibility is there. If it is not occurred, allosteric site, certainly it can be used. That is, I cannot give more actually certain unequivocal term the answer to this question. There are some possibilities.
Thank you, sir. One last question I will take. How does the molecular evolution of enzyme target interfere with the effectiveness of a drug?
No, within one's evolution takes place because as per my understanding, once the human being is developed, the evolution do not take place. From one, actually, the human being, we have reached a particular stage. So we are talking about that particular phenomena. We are talking about actually the evolution, means the word evolution, if you are using it, that particular thing is a primitive stage to another actually the stage. So then we have to find out a new drug. So the enzyme, if it is actually there. Now the within actually lifespan, if the enzyme is modified, means either it is unregulated or it is not effective. The evolution word may not necessarily be used. I somehow am not clearly understood point. Evolution is across actually generations when it is actually evolution. And within one actually lifespan, if there are any changes in a particular thing, so that may be different from one generation. Actually, the pharmacogenetics, when enzymes are actually deficient in some people. Say, for example, these are all actually examples what we give. Mendelian laws. In Mendelian law, whenever we talk about a single gene controlling actually one particular enzyme, acetylcholinesterase and pseudoacetylcholinesterase. So in this particular case, if the in one particular disease the enzyme is knocked off, so that is actually pharmacogenetics. Now pharmacogenomics means multiple genes are actually affected in the different people. So naturally that is pharmacogenetic way the enzyme is actually changed from because of the generation to another generation, and the Mendelian laws, if it works, then actually if they are deficient, that is what exactly when we talk about actually short-acting succinylcholine effect, which is the neuromuscular block agent, and in patients who are actually deficient of pseudocholinesterase, this particular thing can actually produce adverse events, so unless if it is tested. So that way the enzyme actually evolution or modification because of genetics can occur. That is what I can explain. If I do not know whether I am satisfying the question or not. Okay.
Thank you, sir. So on behalf of entire DDH training team and our training team chairman, Dr. G. Narahari Shastri, sir, we thank you once again for accepting our invitation and sharing your vast knowledge with us. We are very much privileged to have you. We are very much overwhelmed to have you in our DDH training program, sir. Thank you so much for joining us. So we'll wind up the session here, sir. From 4 o'clock, vertical 2 will start.
Okay, thank you. Okay, thank you.
Thank you, sir.